Welcome back to Pre-Lab with Mr. Van Dyke, Pre-Lab Round 2. So we are pre-labbing our enzyme lab. This is close to the end of the packet. The start of it looks like this. There are pre-lab questions, and you will need to record what we're doing somewhere. So you can add the procedure directly onto here as we go through, or you can take separate notes, whatever you'd like to do. I will need to see these before you start. And if you have no idea what's happening or what's going on, you won't get to participate in lab. And that's really no fun. So we're going to be working with enzymes and milk and all sorts of fun things this time. So we're going to run through the materials really quickly here. Things you'll have, this is a glucose test strip. This tests for glucose. It's not just a clever name. So this shows if there is the monomer glucose, which is a monosaccharide present in liquids. Oftentimes it's used for urinalysis for testing to see if anyone possibly has diabetes. We'll also be using lactase. This is an enzyme in pill form. This is available in pills because some people are lactose intolerant. So we have lactase to help break that down and so they can digest it easier. We will have sugar water, glassware, beakers, test tubes, etc. Test tube. We will have our friend the pipette. This is NaOH. This is a base. A base is the opposite of an acid. So if you think of lemons, stomach acid, you go the opposite way on the scale that we measure this. That is a base. This is NaOH. You will have Morton pestles. So you will grind up the pills using a Morton pestle. This is what they used to use in apothecaries as well as current pharmacies nowadays. So you just use this and kind of grind it. So that's really it for the materials for the most part. So now this is the part where you start adding and writing down the things you need to do. So one thing you need to do is fill up a beaker with 100 milliliters of water. You will receive half of this enzyme pill, crush it up, and you'll make a solution. You just crush it up and let it dissolve in there. That is your lactase solution, and whenever we're talking about lactase, I'm referring to that solution. I'm not actually going to make it because lab materials are limited, so you need to make sure that you use them responsibly. The next part, we'll run through each test, but they're basically all the same. You'll use 2 milliliters of all liquids for all tests. So for example, the first test is milk and water, and I don't have milk to show you because I don't want to go bad over the weekend. But you'll use 2 milliliters of water and 2 milliliters of milk for your test. So just see what that looks like. These pipettes are 1 milliliter, so you will take 4 squeezes of the pipette. So this could be your 2 milliliters of water, and we'll pretend this is milk. We will add our 2 milliliters of milk. So you should have around this much liquid in your test tube. So now that we have our milk and water solution, we want to test it to see if glucose is present. So give this about 30 seconds to a minute to mix and dissolve into one another, then add your glucose test strip. This needs to sit in there for about 30 seconds. If it changes colors, then there's glucose in there. If it doesn't change colors, and there's a bottle here I have that you can look at to test against, there is no glucose present. I think I said that right. So if it changes colors, there's glucose. If it doesn't change colors, there's no glucose. So that's it. Run it for 30 seconds, clean out your test tube, or use another test tube for your next experiment. So this is just water, so I can empty it back in the same container. So you do the same thing for the next one, except you will use 2 milliliters of milk, and then instead of water this time, you'll use your lactase solution. So the solution you made using the pill and the 100 milliliters of water. So 2 milliliters of milk, 2 milliliters of lactase, add your glucose test strip. So exact same thing. So 2 milliliters is kind of the magic number minus your initial 100. Moving on, milk and boiled lactase. So we'll also be using the hot plates. Remember, it's hot, no. So be careful around them. So boiled lactase, 2 milliliters of boiled lactase. So you will take a test tube and add 2 milliliters of lactase to it. So this is your lactase solution. You are adding 2 milliliters of lactase. So we're doing nothing with milk. No milk. No. We put this into our hot plate. It boils, so we let it sit for around 5 minutes. Now that it's been boiled, now we add the milk, because we just want to see what happens to the enzyme if we add heat. So we have our 2 milliliters of lactase, we add our 2 milliliters of milk. So this is all in here, and again it's just the boiled lactase and milk. We use our glucose test strip, we see what happens. On to the next one. Milk and base lactase. Same idea. You only have a test tube with lactase in it at first. So we have our 2 milliliters of lactase. We take 10 drops of our base. So 10 drops, this will change us from a pH of 7 to a pH of 10. And that's a pretty big change. So we will add 10 drops of that to here, and then we add 2 milliliters of milk. So we'll pretend that we added our 10 drops. So we'll pretend this is milk. We add our 2 more milliliters of milk. Should again be around this high. You know, with the 10 drops of base, it'll be a little higher. Glucose test strip for 30 seconds. The next test is sugar and water. So this is a sugar water solution. So you add 2 milliliters of this. You add 2 milliliters of just regular water. And same idea, add your glucose test strip, wait for 30 seconds, see what happens. Our final one is sugar and lactase. So 2 milliliters of sugar, you add it to your test tube, and then you add 2 milliliters of your lactase solution. So the solution you make, you add 2 milliliters to. And then, glucose test strip. So really, very straightforward, it just can be tricky. If you add the milk and boil it, and add lactase all at the same time, you boil it, it's not going to give you the correct results. So the ones to really pay attention to are the boiled lactase and base lactase, to make sure that you add those ones first. After you complete the lab, there are post-lab questions. So all through here, there are post-lab questions just to test what we've learned about enzymes. And again, the big idea is to get an idea of what's happening inside of our own body. So all these things are happening inside you right now. If you drink milk, your lactase enzyme is taking care of it. If you're lactose intolerant, then it's not. So this is an example of what goes wrong with that in some cases. So if you have any questions, add them to the form. Make sure you have your pre-lab procedures completed so that way you know what to do, and we'll work through this together. So any questions you have, we will address the day before lab. Make sure you fill out the form. Again, safety concerns, goggles, hot plates, tripping hazards. If you get the base on your skin, wash it off, you'll be fine. If you leave it on, you might get a little irritated, but that's about it. Alright. Bye.